Okay, so welcome everyone to this next installment in our talks for accessible genomics and preparing for our sequencing of SARS-CoV-2 and beyond. So the the origin actually of this name accessible genomics actually came from Ineke. It was like a weird long name prior to this, but this just shows like the longer view of this project. It's like it's not just like for this pandemic, but we want to make genomics accessible, you know, through new technology, through a portable lab. So Inike is a PhD candidate from the University of Amsterdam. Her um, research interests include portable genomics, great apes, parasitology, and applied conservation biology. So she'll probably tell us more about her different research interests and about herself as she starts her talk. So let's all welcome Inike to this talk.
Thanks, Khalil. Yes, I love being part of Project Accessible Genomics because in my PhD I've worked with portable genomics, but I've also really thought a lot about how the portability also makes it more accessible because the two aren't synonymous, right? There's also barriers to accessibility that aren't related to portability. So I'm really happy to be giving this talk. And I'll talk a bit about my own research, but also a bit about what others have done. And yeah, so let's see. Yes. So to give a bit of background about my PhD, on which most of this presentation will be based, I am focusing on great apes because all species, all taxa and all species of great apes are endangered or critically endangered. And there are three main threats to great apes, which is poaching, deforestation, but also infectious diseases. And this last threat has been identified most recently. And in 2015, the IUCN has released best practice guidelines for health monitoring and disease control in great ape populations. So there have been some populations in, for example, in Gabon, there has been a local population of gorillas that has been decimated by Ebola. And I think it was in Ivory Coast where there are chimpanzees who have been severely affected by respiratory diseases. So these kind of outbreaks per se have not decimated a species of great apes, but the threat is that if once a disease takes hold of a population, it can spread quite quickly and you can lose a lot of very highly endangered animals at once. So this is why they've developed these guidelines. And that was around the time I started my PhD. So however, if you think about health monitoring and how this kind of happens right now, it's usually you collect data in the field and then you analyze the data. But the collecting the data in the field, in my case, for example, involves collecting fecal samples, storing them, then transporting them either in country to a different lab or outside of the country to a different lab abroad. And then you go to a big lab where you have all these big machines, expensive machines. You need a fully equipped molecular biology lab. And this whole process can take months, but also years. Like most field biologists I know will optimize their field seasons for like a year in the field and then returning, doing all the lab work at once and then doing all the analysis, in which case you can be at least a year, often two, before you have an image on how the health status of your animal of interest was 2 years ago. So in my PhD, we were looking into, is there a way we can reduce this time frame? And we wanted to focus on DNA sequencing for kind of real-time or near real-time biomonitoring. DNA sequencing allows us to focus on yeah, a lot of different things. You guys are genomicists, you know that, but we can look at so many different things if we do sequencing, and we can use non-invasive samples. So for a lot of endangered animals, but specifically great apes, it's really beneficial to use fecal samples because you don't have to disturb the animals. So even if the animals are not habituated, are not used to human presence, you can still collect fecal samples and get a good idea about their health status. So the idea was we can then bring a lab to the field. We can collect samples in a matter of days. We can process them. We can extract DNA and we can do sequencing. And then we can have results in a matter of days instead of months. And then also the idea is that if you're a small scale project, then you can maybe collect some samples on Monday, process them the week, and then go out again the next Monday. And you can kind of do the iterative sampling instead of having to collect everything in bulk and starting from that. So at the basis of my portable lab, there's the MinION nanopore sequencer. Well, we've already talked about that quite a bit in the previous presentations, but yeah, for the portable lab, one of the main advantages is, as you can see, it's not much bigger than a pen. So it fits very easily in your suitcase. And there was even been 1 or 2 occasions where I've kind of thought, oh, where did I put my sequencer? Was it in the office or in the lab? That's a luxury problem to have. And then aside from that, I use the Bento Lab molecular laboratory. So it's a kind of all-in-one unit where you can do the DNA extraction, the PCR amplification, and gel electrophoresis all on a mobile lab that's roughly the size of an A4 paper. There's also other companies, like a lot of people use mini PCR as well. They have like independent parts, so you can order a small PCR, you can order a portable gel electrophoresis unit. So there's different ways in which you can take the whole molecular lab out to the field, but this is the one I used. And then in the last few years, there have been many people that have been finding applications for these for this portable lab. So there have been completely off-grid expeditions to the Arctics, to the polar regions. Obviously, it's been used for outbreak surveillance, which we were doing with Project Accessible Genomics now as well. I've used it for parasites. People have used it for plant pathogens. People have used it to determine the sex of primates. Lots of people have used it for species identification on site. And also there have been some publications about teaching and capacity building, because obviously with this we can now bring labs to regions where they previously weren't, and we can train people, which so I just, the way I kind of built this presentation is that I wanted to go through 3 case studies to kind of highlight a little bit what's possible after the pandemic. I'll mostly focus on my own work with the parasites just because I know most about that, but I'll also highlight the work of Boykin et al. For plant pathogens and the recent work of Chang et al. For species identification. So for my work, as I told you earlier, I focused on great apes, and we focused on great ape parasites. So last year, that was 2019, I went to Indonesia to the Sumatran Orangutan Conservation Program. And I went there to train their veterinarians. I managed to pack my whole lab into 1 big suitcase. And as you can see, my work-life balance was really good because the big suitcase was for work and the tiny one was for my life stuff for a month. So that was ideal. But I looked at nematodes as model organisms. Like I told you before, Ebola or respiratory viruses are really the main threat for great apes. However, sampling a dead animal, for example, for Ebola, involves a hazmat suit like that, and involves a safety level 4, a lab with a lot of precautions, which is logistically very complicated. Whereas if I focused on nematodes as model organisms for epidemiology and parasitology, then we could just do everything in a normal lab, and we could just take minimal precautions. And that just made life a lot easier. So we could validate that the system worked. And as I said before, because we're focusing on DNA sequencing, once we've got something that works, you can very easily adjust a few parts and then you can apply it for other questions. So yeah, I went to Indonesia mostly for capacity building of the veterinarians at Sumatran Orangutan Conservation Program. These vets have a fully equipped veterinary lab to assess the health of the orangutans, but they don't have access to molecular biology, and they do some sequencing every now and then, but they have to send the samples away. And I don't know how it is in the Philippines, but in Indonesia, it's really difficult to send samples even from one island to another one. So it involves a lot of permits and a lot of time. So if they can do it in-house and they can collect a few samples from orangutans over a month, for example, and then spend a few days to sequence them on site, that's highly beneficial. So that's what we were focusing on. And the pictures I will show are from this training, but the results I show are from my first paper. So we collected fecal samples from the orangutans, well, the animal caretakers collected them, and we, because of the stage of my research, we mostly just tried to isolate nematodes from feces and then extracted DNA from that. So the vets identified them morphologically, and then from individual worms, we extracted the DNA. And we did PCR, so we could do all of this on the bento lab. I used the DNA extraction kit with spin columns, so I had to adjust the protocol a little bit to accommodate the bento lab. And then once the DNA extraction happened, we did a PCR. The bento lab can fit 32 samples in the PCR. And then we did gel electrophoresis. We used a little camping stove to melt the agarose. And I've actually found the bento lab to be very cost efficient because your gel is teeny tiny, so you don't use a lot of stuff for it, but you get really clear bands, as you can see on the bottom right, whether your sample works or not. And then after we found out which samples worked on the PCR, we prepared those barcodes for the amplicons for sequencing. And we could also prepare the entire library for the MinION on the bento lab. And then we had the MinION with the Minute, a small mini computer, but we also had a laptop on which we could have ran it. And we did the sequencing on site. That was us looking very happy when the first sequencing run was actually on its way. And then for the bioinformatics, I focused on the on-track pipeline. To be fair, I didn't do that we didn't do that with the vets in Indonesia because I think either the paper wasn't published yet or I was just too focused on finishing my stuff for going to Indonesia, and then I didn't manage to I didn't run it on site, but you can. This pipeline, you can completely download everything you need and you can just run it on a normal laptop. So this is kind of what I did. They kind of describe it as a Pokédex for your species. You can just put your samples, your data in the pipeline, and it kind of clusters, reads, and then calls a consensus and polishes the consensus that you can then compare to databases. So these are the results from my first paper, from which I validated the method with 4 known species of nematodes, Anasaka simplex, Panagrellus redivivus, for example. I looked at the 18S gene, and we got a 99.9% accuracy compared to Sanger sequencing for these two. And for the other 2 species, one was Turbotrix asaeti, we got 100% accuracy with the same primers. And then we also looked at C. elegans. It got a lower accuracy, but that was mostly because I used a very old flow cell. It had been used for 26 hours before, and it was more kind of to validate if I could do DNA extraction and library preparation on the bento lab. Someone reanalyzed my data and got a 99.9% accuracy with slightly different bioinformatics. And also the C. elegans used a different primer pair. So it's something to with such a big phylum as Nematoda, you kind of have to sometimes use different primers to get all the species. But yeah, we were really happy to see this, that the high accuracy for amplicons is there, and we can reliably use it for nematodes. So then we obviously looked at it from a parasitology kind of point of view, but there has been this research by I see I named the wrong year. It was in 2018. It was published. This was a study by a big collaboration of Ugandan, Tanzanian, and Kenyan partners. They looked at cassava virus. So cassava is a agricultural plant that's that's kind of a staple food in a lot of African countries. So farmers are heavily reliant on their plants, and if these plants have a virus, sometimes you can't even really see. Sometimes you can see it clearly, like you can see here on the leaf, you can see a pattern that indicates the virus, but also sometimes I've heard that you cannot really see that anything is wrong with the plant until you harvest it, which is at the end of the harvesting season. And then you have teeny tiny cassava that you can't eat or can't sell. So a whole year's worth of work will be wasted. So one of the things they did here, so they brought their whole portable lab out to the field. They sampled the cassava leaf, they did DNA extraction, they prepared the library, they did whole genome sequencing on the MinION, they performed the bioinformatics in field, found out which virus was present in the field, and then they immediately gave the farmers a resistant cassava variety to plant in their fields. So they knew which virus was present in the field. They chose a cassava plant that wouldn't be affected by that virus, and the farmers could plant that the week after the people left or the day after the people left. So this was amazing. I think this is such inspiring research, and they did that within 48 hours. So a really short turnaround time. So they sampled 15 different locations, and they used, so I used spin columns for my DNA extraction, but they used the cetyl, oh, I didn't practice pronouncing this, the CTAB method. It's written there. And they quantified their DNA with the nanodrop. So this is, yeah, this is one of the things that is also possible with the portable lab, and can have real world implications for agricultural practices. And another study that was recently published was from Chang et al. In Singapore, and they looked at marine metazoa. So they brought their portable lab, which included the bento lab, and they brought it on board of a boat to study marine metazoa. And that this was mostly focused on species identification. So they collected samples via scuba diving, did the DNA extraction and the gene amplification through PCR. All of that on the boat that was moored. They pulled PCR products, prepared the library, then did MinION amplicon sequencing, like I also did for the parasites. And I believe they did all of that on the boat. It wasn't very clear where they did the bioinformatics, but they then also identified the species quite accurately. They also found 70 MOTUs that weren't really, that were novel. But yeah, they had, one of the things that I really liked about their research is that in my research, the most samples I could do at a time were 6, because the bento lab has a small centrifuge and it only fits 6 Eppendorfs. And I used a spin column DNA extraction method, but they used a DNA extraction method that was only needed 2 lysing steps, and they could use the thermocycler of the bento lab for that. So they developed a workflow that you can process 32 samples at a time with. So it's a higher throughput than what I had, and probably kind of the highest throughput you'll get on a portable lab. And their sample to sequence workflow was less than 9 hours. So I also thought this one was worth mentioning. So there's plenty of other work. I just didn't want to overwhelm everyone with the 10 different studies. So these is just a highlight of what's possible for different areas of interest. So I also wanted to discuss the advantages and disadvantages a bit of portable genomics. So obviously the portable thing, I don't think I really have to sell that to you, is that you can do molecular biology at atypical sites. And there are lower startup costs than traditional molecular equipment. Also, from my experience so far, there's less maintenance or servicing of the equipment. Like the Mini doesn't need any servicing. The Bento Lab, once I had a little problem with the lock and I could just grab a screwdriver, open it and fix it myself, which I think is ideal. So I've heard so many stories of really expensive equipment that's been donated and then is in a corner gathering dust because it needs to be serviced every year. And that's talk about accessibility. That's not really accessible in my book. So I think that's really an advantage. And yeah, for me, it's based on my personal experience. It's just a full molecular biology lab. You can do whatever you would do on your traditional equipment. But then on portable, and there's some people have asked me and kind of seem to be worried that, for example, the Bento Lab, because it's portable, produces lower quality results or things like that. But in my experience, that's absolutely not the case. It's just molecular biology equipment that you can use like the traditional equipment. Then another advantage compared to the long-term time frame is, so when I made this slide, it was based on my own work, and nematodes are a bit different because they have a cuticle, very hard skin that takes a long time to break down. So it extends the DNA extraction protocol. But based on my own work, you can go from the sample to having done all the bioinformatics and you're having your species identification already in 3.5 days. And that's with a sample that takes a long time to process. And as we just saw with Chang et al., they had a sample to sequence workflow of 9 hours. And if your bioinformatics only takes then a few hours, then you can get everything done in a day. And I think that's, for a lot of samples, that's realistic now. So there are some time estimates here. I'm not going to go through them in detail, but of course this assumes that the people who are doing the molecular biology are highly skilled, and this assumes that the bioinformaticians that are analyzing the data are highly skilled, because otherwise this will definitely take longer. So in the beginning, when you're doing the portable sequencing, it really takes longer. So then I think some of the disadvantages and bottlenecks are also important to discuss. And the first thing I kind of start with is decide whether you need genomics to answer your question. And I think this is important because, yeah, it's still relatively expensive. So if you can answer the same thing with a microscope, the same question, then that might be worthwhile. Or if you just need, or if you need to get, for example, the sex typing, if you want to know if an animal is a female or male, you can do that with PCR, but you don't need to do the sequencing. So you can kind of think about what your answer needs to be, or what kind of information you need to be to decide whether you need to go all the way to DNA sequencing, or maybe just doing a PCR on site will already give you plenty of valuable information. Because yeah, molecular biology requires skills and all the basic necessities of PPE, pipettes, tips, tubes, reagents. So it's not as easy as just having a microscope and looking at that. But obviously, if you're looking through a microscope, you need a different set of skills again. Proper waste disposal isn't needed if you're in the field. So that's something that needs to be thought of before an expedition or before setting up a molecular field lab. And yeah, there will be a time investment because with the miniaturized equipment, you need to either adjust your protocols. For example, the Bento Lab can only take 1.5 milliliter Eppendorfs. So I tried with 2 microliter Eppendorfs because I'm very stubborn, but it released little pieces of plastic that then got stuck in the lock. So I definitely don't recommend that. And I really hope that Bento Lab will update their beta lab to just accommodate 2 microliters Eppendorfs. But these kind of things, they take time to know, or to pick a DNA extraction protocol that doesn't require spin columns, for example. To get skilled, like fast at doing genomics takes time. To get good at bioinformatics takes time. So if you have a diverse team, as we have in a project Accessible Genomics, then that's amazing. But if you're like me, you have to kind of do everything yourself, then that takes time. And then some other disadvantages are that obviously these tools require electricity. So in Indonesia, for example, they had kind of access to the main grid, but there were very frequent blackouts. So they had a generator for if the electricity cut, but the generator made a lot of sound, and it was quite uncomfortable for the orangutan. So they tried to not use it. So if during the day everyone was there working, then yeah, they would turn the generator on and the orangutans would be awake. But if the electricity cuts at night and you have an experiment that runs overnight, they kind of were like, well, first of all, it's really expensive to run a whole generator just for one piece of equipment. So then we kind of looked at, can we have a car battery? So you have, I think it was called not a CPU, but it's kind of like this thing that you plug your computer in that has like 30 seconds to a minute of electricity before, so that if the power cuts, you can kind of close off your important stuff, save your document you're working on. So we attached the car battery to that, and that worked quite well. So then you have kind of an extra battery pack of the car battery that can run for hours. And yeah, this is known to work. So these kind of things have to be thought of as well in the field. Do solar panels work? How are you going to get your electricity for your tools? And on that same note, it's about refrigeration. This kind of depends on the field site and the length of your stay, because if you're just, if you have a field site that you would go to for a week, and then you can probably get away with using a cool box. But if this is a more permanent stay at the fields, you really have to think about some sort of refrigeration that works long term. I mean, you have DNA extraction kits that can be stored at room temperature. You have lyophilized reagents. But for example, the MinION barcodes with which you can multiplex your samples, they need to be frozen. So these kind of things you have to think of before you go to the field as well. And yeah, because Oxford Nanopore Technologies also has a field kit, but from what I've understood so far, you can't multiplex samples with that because the barcodes need to be stored in the freezer. So there are things like that, puzzles like that, that you can have to, you should, solve before you go. Yeah, and the cool boxes, I already mentioned that. Yeah, another bottleneck I think is it's in my opinion, relatively easy to teach people molecular biology skills. Like pipetting is the most important one. If people can pipette well, and if they can bake, if they can follow a recipe, you can usually get them to follow a molecular biology protocol. But the bioinformatics is, it's just, I think, maybe more complicated. It's just a different type of complicated. And especially because, excuse me, because a lot of the Oxford Nanopore technology bioinformatics tools are so new that they're usually on the command line. So they don't really have a user interface that a lot of people are used to. So just knowing how to code, how to type out all your commands, how to avoid typos, that can really take a long time to master. And also the computing power, a laptop might be enough to run a pipeline like ONTrack, like doing Amplicon-based work. Laptops can be sufficient, but if you want to do whole genome assembly, you really need more computing power, which can be costly and can take days to finish. So that's also something that sometimes people don't really think of in advance. You guys, I think we've thought about that quite well with Accessible Genomics already, but I thought it was worth mentioning, because definitely if the focus of the people who will be using it is mostly wildlife biologists or conservationists or more ecologists, then this is definitely something that people might overlook and think, oh, we can just do this very easily, and then have all the data but don't really know how to process it. And obviously your species identification depends on the quality of the databases. This is what Changata in Singapore found out, that like almost half of their, was it half or 1/3 of their amplicons didn't match to anything, so weren't really submitted to databases yet. If like the risk of only using molecular biology, only using genomics, obviously is that, yeah, you might miss stuff just because it's not in a database. So collaboration with taxonomists are always a good idea. So yeah, also this summer I launched this website, portablegenomics.org. I tried to summarize a lot of the things that I said in this presentation there as well. It has an overview of all the different tech, because there's also qPCRs, for example, that can be brought to the field. So if you want to get like a quick oversight on what kind of other tech is out there, you can go to the website. It's a work in progress. So if you have any suggestions, feel free to reach out to me through the website or on Twitter. To give any suggestions, because I'd love for this to continue to evolve. And then, yeah, I got some really good help from my colleagues, Anne-Sophie Cranchon, Noémie Bonin, and Yannis Sancel, and from the people at SOCP, Jenny Titra and Musa were the vets, and Matt and George Rousson organizers that made my stay there possible. My PhD was funded by the WWF the Netherlands and the University of Amsterdam, and the work in Indonesia was made possible by the Academy Ecology Fund in the Netherlands. So yeah, that's the presentation so far. And are there any questions?
Thank you, Ineke, for the talk. This is really interesting. I think, I'm sorry, I'm not sure if I missed it because we actually have another parallel meeting. I just left
Oh,
the previous meeting. So anyhow, my concern is in terms of on-field taxonomic profiling. Uh, I mean, in terms of specifications for the unit, like laptops are usually all right, or do we need specific RAM requirements? If we're just
yeah,
going to use the ONT pipeline, the what's
yeah.
ONT track.
So I've run it on a laptop with 16 gigabytes of RAM, and that was fine. So I don't know what kind of RAM you have available, and it kind of, I think the ON track pipeline is maybe even possible on an 8 gigabyte RAM. It will just take longer to finish. But like this one, I think it took, I think maybe 30 minutes per sample per amplicon to finish on my laptop. So yeah, I think, and also I'm now for another chapter of my PhD, I'm looking at meta barcoding, and the pipeline I'm running for that also requires at least 16 gigabytes of RAM. So I think that's a good place to be, so to say. I've been able to do a lot of the bioinformatics with that. So yeah, and I also know that, for example, ONTrack was really made with field biology in the background. So it's from Maestri et al., their publication, and they really want this to be able to work in a remote field site where you don't have access to the internet. So they also looked at something that could run on a laptop that is relatively accessible, so to say.
In terms of compiling the local database, like for instance, we are planning to make one for plants, for instance, that we expect to see for our field site next year. For the wildlife training, are there any pipelines that we can use to, compile the database easily instead of like doing it manually?
That's a really good question. I know that there are some databases already available. So you have the Silva databases for 18S, and the Bold databases for barcodes as well. So for example, the meta barcoding pipeline I have, they kind of just use those databases. But for kind of almost semi-automatically compiling your own database, I'm not sure actually. I don't have experience with that, and I do know that I think that's one of the time-consuming parts of bioinformatics, and also because you can also kind of wonder, I mean, the whole reason why you want to build your own database is because you want to be sure that you have kind of the right information. So you can, even if you can automate it, is obviously, I think it will be good to double check that everything is as you want it to be, because so much depends on it, so to say. Yeah.
Thanks.
Do we have other questions from other participants?
I'm on time for the Q&A. Sorry, I was late. We had
Okay.
we had a meeting with the Department of Agriculture just now.
Well, it's all recorded, so you can rewatch the whole presentation. Yeah.
Okay, thank you.
Hello, I have a question.
Yes.
Yes, I'm Joel Tolentino from UP Mindanao also. I'm also connected to PGC as a project staff.
Amazing.
My
Nice to meet you.
question, thank you for that very interesting talk, Ineke. My question is regarding the voltage. How many car batteries do you need to run the machine? The is it bento?
Yeah, so no, not a lot. So I don't know the exact voltage. I can very easily find that after the presentation, but I didn't note it down right now.
But it's not just one.
So yeah, it's the bento lab that you would need, and it's the MinION, but the MinION can be plugged to a laptop. So then that kind of depends on the power requirements of your laptop as well. But the bento lab, I know that there's a fully portable lab workspace, which is made by, it's called Mercury Lab, and it's made by MRI Global. And they have a car battery in their system with a converter. And they say you can run experiments up to 72 hours on one battery. And that's including computer like power, so to say, and including the machines you need to prepare your DNA. So I would say that that's based on their experience, that's probably a safe estimate to have in the back of your mind. But obviously, it kind of also depends. Are you planning to do a lot of bioinformatics on that power as well? Because that requires more from your computer, so that probably requires more energy. So it's a bit, hard to estimate exactly, but I would say that on one car battery, from experience of other people, you can definitely do one full experiment, so to say, maybe even two.
Okay, thanks. How about the gel? Are the gels precast?
No, they are not. I know that, I think for the mini PCR system, I think you can buy precast gels. But I didn't find it necessary. I used I just use agarose with TBE, and the good thing is you only need so little that you can just mix it. I think I only needed half a gram of agarose with 45 milliliters of TBE, one-time TBE buffer, for a 2% gel. And then I used a loading dye, which is called Novel Juice. And you can also, so it's a loading dye, but also the DNA stain at once. And it's very safe. It's more safe than ethylene bromide. So I didn't have precast gels, but because it's so small, it only takes like 15 minutes to set. So I didn't find it a problem to not have precast gels, but.
Okay.
Do you normally use precast gels?
No.
Okay. Yeah.
All right, thank you.
Okay, you're welcome.
Any other questions? All right. So if there are no questions, I hope you guys are okay to continue conversations. A quick project update. I'll stop the recording, and maybe just a quick update on the project. Okay, let me stop the recording.